Welcome to the exciting world of experimental genetics. For centuries, biology was primarily a science of observation, but Thomas Hunt Morgan revolutionized it into a rigorous, experimental discipline. Using the common fruit fly, Drosophila melanogaster, which was bred in simple milk bottles, Morgan and his team at the Fly Room mapped the intricate landscape of heredity. This slide illustrates their groundbreaking discoveries. That genes are located on chromosomes, that linkage groups connect traits, and that chromosomal crossing over disrupts these connections to facilitate genetic recombination. By tracing these microscopic events, Morgan didn't just study inheritance. He mapped it, establishing the chromosomal theory of heredity and providing a detailed understanding of life's mechanisms. Meet the man behind the fly, Thomas Hunt Morgan, who balanced two distinct identities in his early life: the aristocrat and the morphologist. Born in 1866 in Lexington, Kentucky, Morgan came from an illustrious and well-connected family. His lineage included notable figures such as Confederate General John Hunt Morgan and Francis Scott Key, the author of the Star-Spangled Banner. However, Morgan chose to forge his own path as a meticulous scientist. His academic journey began at the State College of Kentucky and continued at Johns Hopkins University, where he earned a Ph.D. in 1890 for his work on sea spider embryology. He later became a professor at Bryn Mawr College. Influenced by his background in morphology, he held a radical belief in sudden macromutations, famously stating, "Nature makes new species outright." Enter Th. Morgan, the rigorous empiricist. This slide emphasizes Morgan's deep skepticism toward the two dominant biological theories of his time. On the left, Morgan questioned Darwinian evolution, doubting whether continuous natural variation could actually lead to the formation of new species. To investigate Lamarckian-style continuous adaptation, his student Fernandez Payne bred 69 generations of flies in complete darkness, demonstrating that their eyes did not atrophy. On the right, Morgan dismissed Mendelian inheritance. He initially regarded Mendelian factors and early chromosome theories as arbitrary, metaphysical constructs that lacked physical evidence. As a strict empiricist, Morgan demanded undeniable experimental proof. He sought a model organism that could breed quickly enough for him to observe and track real-time mutations as they occurred. Behold the perfect specimen, Drosophila melanogaster, the common fruit fly. This overview highlights why this tiny organism has become a fundamental tool for genetic discovery, meeting all the requirements of experimental research. First, let's consider its life cycle. It matures rapidly, transitioning from an egg to an adult in just 10 days, which is much faster than mice or peas. Second, regarding its fecundity, a single pair of fruit flies can produce thousands of offspring, providing a massive statistical sample size for experiments. Third, in terms of its karyotype, Drosophila melanogaster has a remarkably simple genetic structure, with only 4 pairs of chromosomes per nucleus. Finally, the logistics of breeding these flies are highly efficient. Thousands can be housed in small glass milk bottles, each sealed with sponges, and they can thrive on an economical diet of porridge and bananas. This makes them a geneticist's dream organism. Welcome to the legendary Fly Room. This slide highlights the space and network that revolutionized genetics at Columbia University between 1910 and 1928. On the left is room 613 in Schermerhorn Hall, a dusty, cramped 16 by 23-foot lab cluttered with milk bottles. Here, Thomas Hunt Morgan tracked data on scraps of envelopes adorned with squash flies, and famously resisted adopting new technology, such as binocular microscopes. On the right is the secret to their success. An egalitarian team network. Led by Morgan, known as the boss, this open ecosystem thrived on the brilliance of Alfred Sturtevant, the mapping genius, Calvin Bridges, the meticulous experimentalist, and Hermann J. Muller, the tense theoretician. Breaking away from rigid European hierarchies, credit for discoveries was given to whoever conducted the experiment. This is the moment everything changed: the discovery of sex-linked inheritance. In step 1, we start with the catalyst. Morgan finds a single male fly with mutant white eyes instead of the normal wild-type red eyes. In step 2, he breeds this white-eyed XY male with a red-eyed XX female. The resulting F1 generation exhibits 100% red eyes, demonstrating that red is the dominant trait. The real surprise occurs in step 3. When Morgan breeds the F1 offspring, the white eye trait reappears, but only in the grandsons. This led to a significant revelation. Because males inherit their single X chromosome from their mother, the eye color trait must be located on the X chromosome. Genes were not just abstract concepts, they were tangible, physical objects. This slide introduces a significant departure from classical genetics, the concept of linkage. As more mutations were discovered, Morgan realized that Mendel's law of independent assortment was not entirely accurate. The mechanics behind this explanation are straightforward. Genes that are located on the same physical chromosome do not assort independently. Instead, they are linked and tend to be inherited together. To illustrate this concept, the slide uses the analogy of a railway system or a necklace. Genetic traits, such as eye color, body color, and wing shape, are arranged linearly on a physical chromosome, functioning like stations along a single railroad line or beads on a string. A new puzzle emerged regarding genetic linkage. It wasn't always perfectly consistent. Occasionally, these tightly linked traits would unexpectedly separate. To explain this anomaly, Morgan identified a process he called the mechanism crossing over. Drawing on cytological observations of meiosis, he proposed a dynamic event in cells. Paired homologous chromosomes twist together, break apart, and physically exchange segments of their genetic material. This behavior led to the rule. The physical exchange of genetic material during crossing over directly disrupts linkage between genes. By actively swapping matching segments, chromosomes recombine maternal and paternal traits, creating immense genetic diversity that drives evolutionary change. This slide presents a significant breakthrough: the creation of the world's first chromosome map. The insight emerged when Morgan theorized that the strength of gene linkage is directly related to physical distance on the chromosome. His student, Alfred Sturtevant, realized that this concept could be calculated mathematically. The mathematics is elegantly simple. If chromosomal breaks occur randomly, genes that are farther apart are statistically much more likely to be separated by crossover events than genes that are located close to each other. Thus, a greater physical distance corresponds to a higher chance of crossing over. The result of this research was revolutionary. By translating crossover percentages into physical map units, the team successfully charted the X chromosome of Drosophila melanogaster, marking specific loci from yellow body to bar eyes. They had effectively mapped the geography of life. This slide illustrates the paradigm shift in biology, highlighting the differences between 19th-century morphologists and Morgan's genetic paradigm, which shows how biology underwent a permanent transformation. First, the focus in biology shifted from purely descriptive and morphological observations to an experimental, quantitative, and predictive approach. Second, the understanding of heredity evolved from abstract, invisible Mendelian factors to concrete physical genes located on specific chromosomes. Third, the perspective on evolution transitioned from speculation about sudden macromutations to tracking actual genetic mutations and recombinations across generations. Finally, the concept of the gene itself was redefined. It was no longer an abstract theoretical idea. Instead, it became a physical, mappable coordinate, behaving like pearls on a string. With these changes, biology officially entered the modern era. This slide illustrates the westward expansion of modern genetics, highlighting the transition from the East Coast to the West Coast. The Move, 1928, recounts the story of 62-year-old Thomas Hunt Morgan leaving New York and the confined Columbia Fly Room to establish the brand new division of biology at Caltech in Pasadena, California. The migration shows that Morgan did not make this move alone. He brought along trusted colleagues from the Fly Room, such as Sturtevant and Bridges, and recruited top-tier experimentalists like Dobzhansky and Beadle to join him at Kirchhoff Laboratories. Finally, the philosophy outlines Morgan's vision. With generous endowments, he developed a world-class program that prioritized physical chemistry and rigorous experimental research over traditional descriptive morphology, ultimately shaping the future of biological science. Thomas Hunt Morgan was awarded the Nobel Prize in Physiology or Medicine, the first ever given to a geneticist, for establishing the chromosome theory of inheritance. True to his workaholic nature, Morgan skipped the 1933 ceremony in Stockholm due to his intense research schedule, but finally attended the event in 1934. The right side reveals the ledger. Prize sharing, which uncovers the human drama behind the science. Staying true to his collaborative lab ethos, Morgan shared the prize money with his long undercompensated flyroom colleagues, Alfred Sturtevant and Calvin Bridges, as well as his own children. However, tensions remained high. Morgan pointedly excluded Hermann J. Muller from the financial sharing, casting a lingering shadow and highlighting the deep, bitter personal divides that quietly fractured the legendary flyroom era. Welcome to the ultimate proving ground of modern genetics. This concluding slide illustrates how Morgan's dedication to experimental verification forever changed the field of biological science. Under the section Nobel Lineage, we see the enduring legacy of his Caltech department, which produced numerous future Nobel laureates. Among them were Hermann Muller, recognized in 1946 for his discovery of X-ray mutations, and Edward Lewis, who mapped Hox genes. Finally, the modern fly highlights the ongoing significance of Drosophila. Today, over 50,000 scientific papers reference this humble insect. Remarkably, about 75% of human disease-related genes have direct counterparts in the fruit fly, making Morgan's classic model organism an essential tool for biomedical research into Alzheimer's, Parkinson's, and cancer today.